Meet Pete. Pete is a junior scientist at a typical CRO. He has just been asked to lead a Phase 1 first in-man clinical trial. To help him, Pete receives an investigator's brochure, an IB, from the sponsor. In this IB, all the preclinical research is bundled together. This massive document contains hundreds of pages loaded with information about chemistry, toxicology, pharmacology, and loads of other preclinical information. Pete will need to refer to the IB before, during, and after his Phase 1 trial. From this sea of data, he will somehow have to determine a safe but effective dosing regimen. But where does Pete even start? If he chooses a dose that's too low, he won't learn the possible benefits of the test drug. If Pete chooses a dose that's too high, his patients could develop severe side effects. Pete starts by reading the IB. He spends hours and hours trying to find the information he needs. Some doses are given in molar units, some are given in micrograms per kilogram, and others are given in milligrams per kilogram, milligrams per milliliter, milligrams per day. All of this in different species and genders, sometimes experiments reporting concentration, sometimes in half milliax inhibited concentration. There has to be a better way. Pete, I think I can help. There's a new tool available that can give you a clear overview of all the data in an IB. It's called Derisk Analyzer. This free-handed tool quickly and easily organises the available data into a clear table, which is required by the EMA guidelines. It's quite simple. I'll explain how it works. We'll use Derisk Analyzer to extract species, dose or dosage, and blood or plasma levels. Each entry is assigned a coloured tag representing safety. So, for example, if a study with rats yielded safe results with no side effects, this is labelled green for safe. On the other hand, if a study with monkeys showed severe side effects, this would be labelled red for potential toxicity and or death. Using this tool, Derisk Analyzer allows you to quickly sort all studies in the IB by dose, concentration, outcome and more, like with graphs of maximal concentration versus dose for different species. Now, with just a glance, you can find the ideal dose of your study. From now on, Pete doesn't need to worry about finding the right dose. He can focus on designing the best trial. To learn more about how Derisk Analyzer can help you, visit their website at www.ib-derisk.org.